And so let's take a look at your activity one, instrumentation. Okay, and so you have here, and so under this, and so we'll be also talking a bit of your laboratory safety with under this presentation. Okay. And so your clinical centrifuge. Your clinical centrifuge is a laboratory equipment designed for separation of components in liquid based on their density. And so probably you have a solution. Okay, and so but then you're trying to separate the components of that. And so based on their density. And so you have here actually an illustration of a centrifuge, and so with the essential parts here. Okay, and so of course please also note the use of your tachometer in your clinical, the tachometer that is present in your clinical centrifuge, no? And so with this, in relation to clinical microscopy, and so this one is used in recovering solid materials from suspensions. And so example, of course, is the microscopic exam of your urine. Okay, and so we obtain a sample. Of course, with that, we place a sample from your urine container into your test tube, and then we centrifuge it. And so we centrifuge the urine, and then we decant. And so you have actually a sediment present. We decant the supernatant, and then what is left is the sediment. And so the sediment then is then examined, and so under the microscope. Okay, and so that is for the use of your clinical centrifuge in the clinical microscopy section. Okay, and so of course you have other uses also with that. Moving on, you have your urinometer. And so your urinometer here accordingly is a weighted glass float with a long, narrow calibrated stem. And so this one is your stem here. Ayan, so that is the stem. Okay, and so your urinometer accordingly will sink and will displace a volume of liquid that is equal to its weight. Okay, and so you have here, and so for the illustration here, and so you have actually it is, uh, of course, still you will calibrate it at using a distilled water to give you a specific gravity of 1.000. Okay, and so of course you have here it should be free floating, and so it should not touch the walls of the test tube. And so of your test tube, and so with such again, and so the urinometer will sink and will displace a volume of liquid equal to its weight. And so with that, of course, you already have your specific gravity with that. Okay, and so that is for your urinometer. And so you have here your urinometer is otherwise known as hydrometer, and so it is otherwise known as a hydrometer. And then you have your hemocytometer. And so your hemocytometer here, and so this one is a specialized slide used for cell counting with a microscope. Okay, again, your hemocytometer is a specialized slide used for cell counting with a microscope. And so with this, of course, you have here, you already know this one. You have a two counting chambers present here, one and two. And so as for cell counting in clinical microscopy, and so cell counting is done. And so when you do your sperm analysis, and so to give you a sperm count. Also in cell count, and so when you perform your cell count, differential count of your other body fluids, like for example, you have a turbid CSF, and so you are required to do cell count. And so cell count, you're going to count the cells present. Other than that, and so your other body fluids, examples again, you have here your synovial fluid. And so you need to do such, okay? And so, of course, how are you going to do this is you're going to charge. And so you're going to charge your hemocytometer. And so the ones illustrated here is an improved Neubauer uh, hemocytometer. Okay, and so you're going to charge. And so that is why you need your WBC thoma pipette. And so you need your WBC thoma pipette. And so to dilute, okay, and so to dilute your to prepare a dilution of your sample. Okay, and so to prepare a dilution of your sample, and so if you have a turbid other body fluids, and so for cell count, then you need a thomapipette, probably a WBC thomapipette to uh, do a dilution, and so to dilute. Okay, and so with that, of course, you're going to do the dilution and then you charge. Okay, and so china-charge po siya. And so the term we use in hemocytometer is charge. Hindi po siya drop. Okay? And so you charge, you have actually a small slit here at the side, and so that is where you're going to charge. And so after you charge, then you place the cover slip. And so you place the cover slip, and then you are good to do your cell count, differential count. And so this is the structure of this one. Okay, and so this is the structure. And so enlarge, if you try to enlarge the one counting chamber, and so ganito po ang itsura. Okay, and so this is again your hemocytometer used in cell counting. And so cell counting. And so this one also, your hemocytometer also is done, is used in the hematology section. Ayan. Also to do manual cell counting. Again, to do manual cell counting of RBCs, WBCs, together with, together with, together with your platelets also. And so, but then, and so uh, in case of clinical microscopy, and so you're using this one to count cells present in your other body fluids, like you have your CSF, synovial fluid, uh, and of course the rest. Okay, you have there. Moving on, you have your refractometer. Your refractometer, and so it utilizes actually your refractometry. And so your refractometry determines the concentration of dissolved particles in a specimen by measuring refractive index. And so you have there. And so comparing it actually to your uh, urinometer, and so urinometer is a direct, and so is direct measurement. And so comparing it to this, and so you need to measure your refractive index here. And so this one is an illustration of your refractometer. And so you have here your sample plate, or your sample plate. And so you have here, that is your eyepiece. Okay, and so this is your eyepiece. Okay, and so dito po tayo sumisilip na part. Okay, and so with this, and so you have here for your, this is where you place, of course, your sample, your specimen. Okay, and then of course you have here your scale as such. Okay, and so you have there for your refractometer. And so it is actually, yes, and so you have here for your refractometer, and so that is calibrated at 1.00. And so for your distilled water, using your distilled water, and so you calibrate your refractometer, and so using your distilled water. And so the calibration of your refractometer should be done daily, and so if you are using such in the laboratory. Okay, and should be uh, at 1.000. Okay, and so with that, and then of course that is calibrated at 1.005, graduated at 1.005, 1.0010, 1.0015, and the rest. Okay, and so you have there. And so but then careful, and so because other uh refractometers, and so they use actually uh they have actually serum uh specific gravity. And so but then of course you check, and so you check the label. And so if you're using the scale for urine and or for serum. Okay, others also, they have graduations at 0, 5, 10. And so but then with that, and so you start at 1.00, and so per uh per what they call this, per line, and so per, yes, per graduation, and so you have there your, you add 0.005. Okay, and so with that, it will also follow 1.000, and so next will be 1.005, then 1.0010 also. Okay, and so you have there. Moving on, and so let's take a look at your microscope. And so others, of course, you have here other instruments that are utilized in the laboratory. Of course, you have there your glass slide. And so your glass slide, and so that is, of course, where we place your urine sample, and so the urine sediment for microscopic exam. And then uh, also, and so other than the use for microscopic exam in urine, and so we are also utilizing glass slide to stain uh, samples, like let's say for example in cell count, diff count, when we do differential count, and so we need also to stain. And then of course, and we utilize your glass slide to do such, and so to prepare a smear, and then we stain. And then other than that, and so uh also in sperm ana, and so we are utilizing also glass slide to prepare smear, stained smear of such, and also to use to visualize your morphology and the motility of your sperm. Okay, and then you have your test tube and test tube rack, of course, and so you are utilizing your test tubes, and so for centrifugation purposes, for centrifuging purposes also. And then for your dropper, or your for your pasture pipette, and so of course you are utilizing such to transfer samples. Okay, and so if you need to put a drop of a sample in a glass slide, and so as such. Okay, and so moving on, let's take a look at your microscope. And so of course your microscope, this one is an instrument used to view an object that is too small to be seen by the naked eye. Okay, your microscopy, as what I've said last time, and so that is the technique of utilizing the microscope, and so that is the technique in that is your technique, or that is the science actually of investigating small objects, and so with the use of your microscope. And so as such, uh you have here an illustration of a conventional compound binocular microscope. And so with this, you have here your eyepiece, or you have your ocular lens, or your eyepiece. Of course, that is where you, what you call this, that is where you view. Okay, you have your diopter adjustment. And so this one is an adjustment to correct a difference in the vision, okay, of vision. And then you have your revolving nosepiece, and so of course it houses your objective lenses, and it can be revolved, no, and so for uh yes, and so to change your objective, and so if you want to change from low power to high power, then your revolving nose piece will do that for you. Okay, and then you have here, of course, you have your objective lenses, you have your stage clip, together with your stage, your mechanical stage, your diaphragm, your condenser, your light source, your rheostat, and so your brightness adjustment, your stage controls, and so your X and Y axis knobs, your fine adjustment, and then your coarse adjustment knob, and then your mechanical stage. And so if you want to add, you have your draw tube, body tube, or the body, and so those items. Okay, and so with that, let's take a look at your parts. Let's take a look at the parts of your microscope. And so you have here your eyepiece, that is the lens where the viewer looks through. Okay, and so that is the lens where the viewer looks through. Okay, and so with this, and so it further magnifies the image. And so last time in the lecture, and so I told you that a microscope has two lens system. And so it has a two lens system. One, your magnifying lens system. You have here your the first one, the primary one is your objective lenses, and the second one is your eyepiece. And so your objective lenses, and so they are utilized for initial magnification of the object. And then your eyepiece, and so of course it will further magnify the image that is uh, that is processed by your objective lenses. Okay, and so usually your eyepiece, and so it is a lens, it's capable of magnifying an image, and so with that, a typical magnification that is present in a compound microscope in the eyepiece is at least 10 times. Okay, and so at least 10 times. Okay, and so moving on, you have your objective lenses. And so your objective lenses, this is housed in your revolving nose piece. And so with this, actually, you have actually three to four objective lenses housed in your conventional binocular microscope. And so with this, and so for you have your scanner, your low power objective, your high power objective, and your oil immersion objective. And so for the magnification, you have your low power objective that is at 10 times. And so your high power objective has 40 times, and then your oil immersion objective has 100 times. Okay, and so when you say total magnification, and so you multiply your objective lens, uh, the magnification in your objective lens, multiply it by your eyepiece. And so let's say you are using low power objective. Okay, and so the magnification of your low power objective is 10. And so you multiply that by your eyepiece. And so that is a 10. To give you 10 times 10 will give you 100 times. And so that is the total magnification. Okay, and so when you change the objective, and so to oil immersion, then you multiply your 100, you multiply it by 10 to give you 1000 as the total magnification. Okay, and so with that, for your objective lenses, you have actually color bands present in them. And so for your scanner, you have red. For your low power objective, high power objective, and oil immersion objective, they have yellow, blue, and white respectively. Okay, and so moving on, you have your condenser. Your condenser here, and so it focuses light from the illuminator to the specimen. Okay, and so your condenser. And then your iris diaphragm, and so it controls the amount of light that reaches the specimen. And then your revolving nose piece, and so it houses the objective lenses. And so it houses the objective lenses, and so it can be rotated, and so that allows for quick change in objective. And so let's say you are focusing on low power, and then you wanted to see in high power, then you just rotate your revolving nose piece, and so for a quick change in objective. Okay, and then you have your coarse adjustment knob and your fine adjustment knob. And so for your coarse adjustment knob, and so this one is utilized in general focusing of the objective. I mean of the object. And so coarse adjustment knob used for general focusing of the object. And so usually your coarse adjustment knob is used when you are using your low power objective. Okay, and so when you are using your low power objective, and so coarse adjustment knob is used. Okay, and so you have there. And so also, and so your coarse adjustment knob, and so this one is utilized to move your mechanical stage up and down. Okay, and so this one is utilized to move your sample closer to the objective lens. And then your fine adjustment knob, and so it increases the detail of the specimen. And so you already have a coarse focus, then you need a fine adjust, then you need to see the detail. And so then of course you manipulate your fine adjustment knob. And so you have if your course adjustment knob, and so is utilized in conjunction with your high power your with your low power objective, your fine adjustment knob is used when you are using your high power objective naman. Okay? And then you have your mechanical stage adjustment knobs, and so it allows the movement of the slide horizontally. And so you can move it uh, left to right. Ayan, as such. Ayan, so again, it allows the movement of your slide horizontally. Ayan, so your mechanical stage adjustment knob is otherwise known as your uh, X and Y stage knobs. And so X and Y stage knobs. Okay, and so your rheostat. And so this one will control the light intensity. And so it will control your light intensity again. And so you control the light intensity. And then your stage. Of course, your stage, that is the platform where your slide is placed. Okay, and so that is, if you want to add your stage clip, and so your interpupillary distance scale, and so as such, and so are also present in your microscope. Let's take a look at your care and preventive maintenance of the microscope. And so, of course, with this, you carry the microscope with two hands, and so supporting the base with one hand. Okay, and so always hold the microscope in a vertical position accordingly, and so clean optical surfaces only with a good quality lens tissue and commercial lens cleanser, and so do not use solvents, and so in doing such, and do not use the 10 times and then 40 times objective with oil. Okay, and so with oil, and so for your low power and high power objective, that is for dry only, and so dry only. And then of course you clean the oil immersion lens after use, and so you clean after use, of course, because and so you don't want the oil to you don't want it to what do you call this. Yes, and so you didn't want it to be stuck in your uh, objective lens, okay? And then always remove slides with your low power objective raised, and then store the microscope with your low power objective in position and stage centered. And so as such also, and so do not use mascara, and so eye mascara in your when you are utilizing the microscope. And so of course it might destroy yes, it might uh, destroy the lens of your eyepiece. Okay, and so with such, okay, and so of course, and so when such, and so you are needed, and so you always consult your manufacturer, or you always consult your uh what do you call this, your manufacturer's manual also with that. Okay, and so that is for your microscope. Moving on, let's take a look at your standard precautions. And so for your standard precautions, and so they are used for all patient care. And so, of course, this one, they are based on a risk assessment, and of course, they make use of common sense, no, and practices in personal protective equipment that protect healthcare providers from infection and prevent the spread of infection from patient to patient. And so under your standard precautions, you have actually this one, your standard precaution actually is your first tire. Okay, and so first tire. And so with this, you have your first on the list is, of course, you perform hand hygiene. And so as you always, as you know, and so as you know that your hand washing or proper hand washing, and so is the single most effective way in preventing the spread of diseases. Okay, and so with this, of course, you perform hand hygiene. And so patient, in between patient. Okay, and so after handling a patient, And so after handling a patient, after touching a patient, and then before touching a patient. Okay, and so you perform hand washing or hand rub. And then you use personal protective equipment whenever there is an expectation of possible exposure to infectious material. And so with this, of course, since you are working in the laboratory and you're handling infectious material, then of course you are required to wear your personal protective equipment. And then of course with this, and so you in response, I mean in addition to your personal protective equipment, and so of course you are also required to know and so the proper donning and doffing of your personal protective equipment. Okay, and so which is to be put on first and which is the last, and so which is the first to be removed and which is the last. And again, you are required to know the proper donning and doffing of your personal protective equipment. And then you follow respiratory hygiene or cough etiquettes. And so also ensure appropriate patient placement. And so that's one is in cases of the patient needs isolation, needs to be isolated as such, and then properly handle and properly clean and disinfect patient care equipment and instruments. Ayan. And then ensure healthcare worker safety, including proper handling of needles and other sharps with this. And so you have actually others for this one that will include uh, what do you call this safety in handling linen. And so but then in you under your standard precautions, and so you are required to ensure your healthcare worker safety, including proper handling of needles and other sharps. And so with this of course, and so to prevent uh, needle sticks, and so transmission of needle stick, I mean of yes, of bloodborne pathogens, and so those items. Okay, and so that is for your standard precautions. Of course, safety, and so safety is a concern of everybody. And so your safety should be your uppermost priority. Okay, and so it should be your uppermost priority.